those of you walking that way, watch out for the drip from the ceiling. I got hit by it, so. Okay, well, we didn't chase everyone away last week. Let's see how we do this week. I'm going to talk kind of a continuation of what we talked about last week, which had to do with just the sheer mathematical improbability of random mutation causing Darwinian evolution. And we left you last week saying how it's even harder than that. Do a little bit of review here. Always start with this slide every time I do one of these, is that, science is interesting, but science changes. Don't base your faith on what you hear from science, because they'll change it the next day and, remove the foundation of your faith. You don't want to do that. Remind everyone of the famous Time magazine cover from 1966, Is God Dead? There's been a lot of scientific research since that time. It all points in the opposite direction from what Time magazine was intimating, I guess, almost 60 years ago now. Whoop, now I went the wrong way. Okay. Well, Dan, can you hit the down arrow one time? Yeah, so it'll be the lower right-hand corner of the keyboard. And no. Pardon me? I'm sorry? Down? No? Ah, okay. No, we went all the way to the end that time. Okay. I'm going to back up through this. It seems to be... Okay. Let's go to this one. Okay. Wow, technology. This didn't happen last week, but okay. Just remind everyone of these two books, one by Stephen Meyer, the other by Eric Metaxas. The one by Metaxas especially is very accessible and goes over what we've talked about the last couple of times. And down there, there we go. Okay, quick review from last week. We talked about there's information stored inside DNA, and it was shown that that information behaves the same way an alphabet does, and there's certain combinations that make sense, just like certain combinations of letters make sense in the English language, but there's an infinitely bigger set of combinations of letters in English and combinations of nucleotides in DNA that don't make sense at all. And it is such a significant problem is that, scientists have researched this, and they've shown that the probability of finding a functional random mutation within DNA is one chance in 10 to the 77th. And we went through some examples last week of how big of a number 10 to the 77th is. It would be as if you covered the entire surface of the earth with tiny grains of rice, miles and miles thick, and the chance of finding a functional DNA sequence by random mutation is the same as finding one particular grain of rice out of the whole earth covered miles deep in rice. It just doesn't happen. So, and a lot of this we found is based on things really Darwin didn't know when he put his theory together. He thought at the time, and the best science they had was that the smallest unit of life was the single cell. It had a membrane, it had a nucleus, it had goo inside cytoplasm, right? But we now know through the field of molecular biology that the cell is far, far more complex than that. That's where mutation and DNA changes really happen, and they are far more complicated than anything that could have been envisioned back in the 1850s. So, with that as review, I'm going to move on. We're often taught evolution in school, or we hear it in popular arguments with what some people call just so stories. And you've seen charts like this, right? There was some tubby animal with a slightly long snout millions of years ago, and millions of years later, there's another tubby animal with a slightly longer snout, and millions of years after that, the snout gets longer, until the modern day, you've got the elephant, right? Same story with the giraffe. You got something that looks kind of like a horse. He's reaching up in the trees to get food. Millions of years later, he evolves to have a longer neck, and millions of years later, a slightly longer neck, and finally in the modern day, you've got a giraffe. All these little changes give the animals a slightly better chance of survival, and thus, natural selection weeds them out, and you get evolution. Everything here is, again, based on numerous slight modifications over billions of years of time. It is based on the fossil records such that they exist. It's also based on external anatomical similarities. You can kind of see externally what's going on there in the anatomy. Darwin used these because it's the best he had back in the 1800s to explain evolution. And like I said, you still see a lot of this today in textbooks. But as we learned last week, this is not where the changes really happen. It's happening at the molecular, the DNA level. Darwin was honest enough back in the 1800s to make a statement that I've got up there in the lower left-hand corner, where he said, if it could be demonstrated that any complex organ existed which could not have possibly been formed by numerous successive slight modifications, my theory would absolutely break down. So, as you might guess, we have an example. Let's look at the just so story for vision. This is another one based on the same things that the previous just so stories were based on. You'll hear the story of vision described like this. Some tiny creatures are going to have a spot of light-sensitive cells somewhere on them, and this gives them an advantage in survival and gets passed on, and over millions of years, for some creatures, that spot of light-sensitive cells evolves within a depressed region, and that gives them the ability not only to sense light, but to sense which direction it is coming from, so that gives them an advantage towards survival, so that trait is passed on. And then further on, in some creatures, that depressed area gets filled with a fluid, and that acts as a crude lens and gives them a slight advantage in survival, and so on. And it continues evolving until you get the complex eye with an improved lens like higher animals have. Now, this just so story, it makes a certain amount of sense, but it ignores certain things. Like, okay, you've got some light-sensitive cells, so what? They need a nerve to carry the light sensing that they do back to something called a brain, and the brain's got to be able to interpret it. And so, the light-sensitive cells in and of themselves don't do anything, right? What about that? What's going on there? And then further, as you would guess, the just so story for vision isn't really what's going on. We know that it's happening at a much more complicated and lower level. Whoops. And there we go. So, what's really going on biochemically within the cell at, the chemical level, and this is an eye chart, you don't have to read it. The whole point here is this is complicated, right? So a photon will strike a light-sensitive cell. It's absorbed by an inactive photoreceptor protein called rhodopsin, which leads to an activation of another protein called transducin. The activated transducin activates another thing, which I won't even try to pronounce, but I'll call it PDE. The PDE hydrolyzes something called cGMP, which reduces its concentration. The reduced concentration of cGMP closes a channel on the wall of the cell where positively charged sodium ions flow back and forth. After that channel is stopped, the polarity of the cell changes, and as a result, an electrical signal gets sent down the optic nerve all the way to the brain. The brain interprets that signal as vision. All of this is followed by an equally complicated reverse reaction to go back and set that initial inactive rhodopsin protein back to its initial state so that it can receive the next photon of light and interpret it. And all of this happens in picoseconds, and a picosecond is 10 to the minus 12th seconds, or 0.000000001 seconds, right? Now, it seems rather incredible that this could have evolved piece by piece, because if you look at each chemical piece of this along the way, it provides no discernible survival advantage for the organism that developed it. All of these things, and it actually is even more complicated than what I've put up here, would have to show up at once in order to create a survival advantage. And, could this be a direct example of what Darwin himself said would invalidate his theory? There is a name that has been given to this, and it's something called irreducible complexity. And irreducible complexity is often illustrated in the literature over the last 30 years by using the example of a mousetrap. A mousetrap has 5 parts, right? It's got a flat wooden platform base. It's got the metal hammer, which kills the mouse. It's got a spring that puts pressure on that hammer. It's got a catch to hold the spring back. It's got a metal bar that connects to the catch and holds the hammer back when you charge the trap. So the question to ask is, which one part of this could I take away and it would still function as a mousetrap? And the answer is none of them. They all have to be there, or it doesn't function at all as a mousetrap. It has no advantage. Or another way of asking this, how could you evolve a mousetrap one piece at a time, right? You start with a base. What advantage does that give you in catching a mouse? None. You do a base plus the metal hammer. What advantage does that give you? None. You don't get an advantage in catching a mouse until all 5 things show up at one time. Are there examples of these in life, in evolution at the molecular level that they couldn't have known about 150, 200 years ago when they made the theory of evolution? And it turns out there are tons of them, right? Mentioned vision before, and you realize that it is much, much more complex than the just so story that we showed a few slides back. Blood clotting that we're all familiar with. You cut yourself and the blood will clot and form a scab there, but something we never think about, something has to stop that clotting, scabbing action, otherwise all the blood in your body would clot up and you would die. So there's a reverse reaction in there that's very complex as well. There's cilium motion on small bacteria, there's flagella. The point of all this being, if as last week, we found out that the probability of finding a single functional mutation is 1 chance out of 10 to the 77th, which is an unimaginably small number. The probability of finding multiple simultaneous functional mutations all happen at the same time is 1 out of 10 to the 77th times 10 to the 77th times 10 to the 77th, on and on and on. It is such an improbable thing. It's not even worth talking about as being different from 0. It just couldn't happen. So, like last week and all the stuff, why haven't we heard about this, right? Interesting question. Back almost 30 years ago now, there was a biochemist at Lehigh University named Michael Behe. He started looking into this, and he said, well, this is really interesting. I don't see how evolution could have produced such complicated, irreducibly complex structures as what we're seeing here. And he wrote a book about it called Darwin's Black Box. And as part of writing the book, he decided to research what have my peers been writing about this? This is a pretty big deal. I need to understand what other research is out there to test my theory against. And kind of the journal in his field is called the Journal of Molecular Evolution. Began publication in 1971. There have been over 2,000 papers published from 1971 through 1996 when he wrote his book. In his absolute surprise, he found out that there have been exactly 0 papers published on this at that time. It was like nobody was interested in it, and it just blew him away. How could you not be interested in this? This is the biggest thing you could possibly study in our whole field, and nobody's looking at it? Okay, I'll look at it. He published the book, created a big stink, as you would imagine, and he got the same reaction that we mentioned last week. Some scientists went and actually took a scientific approach and tried to show how things could evolve this way, but most gave a lot of ad hominem attacks, and even his own university felt compelled to put something up on their website that said, we do not support his work here. We cannot put ourselves behind it. And as he asked, why can't you cite a scientific paper in support of your position that you don't support me? And of course, there wasn't one. Fortunately for him, he had tenure, so he didn't lose his job. And he kept writing. He wrote a bunch of other books, one called The Edge of Evolution, where he looked at microevolution, and he found at the molecular level, evolution does produce change within a species, but there's a limit beyond which it can't go. It becomes actually harmful to the species. A few years later, he wrote one called Darwin Devolves that showed that as mutations happen within a species, they invariably don't improve the species, they degrade the species, make it less likely to survive. And most recently, just about a year and a half ago, he published a Mousetrap for Darwin, Michael Behe answering his critics. The point being of all this, in conjunction with last week's talk, between the mathematical challenges at the DNA level and the molecular biological challenges, evolution is a theory on very shaky ground, contrary to what you hear by some of the aggressive evolutionists out there that go around promoting this. And, the monkeys we talked about last week and the mousetraps this week, they're not the end of it. There are more and more challenges in the scientific literature out there. There's a field called abiogenesis, which has to do with creating life out of non-living things. And it refers back to a famous experiment done in the 1950s called the Miller-Urey experiment, where they created something like the primordial prehistoric environment, an ocean with the right chemicals in it, an atmosphere with the right composition, and they shot lightning through it, and lo and behold, they created some fundamental proteins and amino acids, and they thought, aha, we've got it. We know how life came from non-life. But since that time, there's been zero progress in the field, and as a matter of fact, everything that's been discovered points the other way. They're farther away than they were in 1952 from figuring this out, although there's still a lot of baloney that gets slung around out there that they do know how this worked. And it provoked a guy named Dr. James Tour up there. He's head of a department at Rice University. He's the world's foremost expert on something called nanotechnology. It's how you assemble machines out of individual molecules. He's actually assembled a working car out of carbon molecules. It's fascinating. He holds 200 patents, published 600 papers. He is the most cited researcher in the world on molecular nanotechnology. And he got fed up with all this baloney several years ago, and he said, stop it, you guys. You have no idea how this works, and I'm calling you out on it. It got so far in 2019, he challenged 30 of the leading scientists who study abiogenesis, life from non-life, who are quite mad at him. He's got his own website and YouTube page where he publishes all the stuff challenging their theory. He said, I'm going to put up 5 questions. I'm going to challenge the top 30 guys in this field. If any one of you can answer any one of my 5 questions on this topic, I will take down my website, I'll destroy all my YouTube videos, I will shut up, and you will never hear from me again. His website is still up, okay? Second thing, something called the Great Darwinian Paradox, for which a Nobel Prize was awarded in 1995. 1995, a bunch of scientists did genetic manipulation of fruit flies. They always do these experiments on fruit flies because fruit flies breed fast and you can go through a lot of generations. And they proved, and won the Nobel Prize for it, what Michael Behe talked about in his book, that the only genes you can vary are the ones that handle minor form and function, like whether you have red hair, blonde hair, blue eyes, or brown eyes. The genes that govern major changes, the kind of thing Darwin said help turn one creature into a new species, invariably you either can't vary those genes, or if you do, you kill the animal. The mechanism just doesn't work. Another thing called the Cambrian explosion has to do with a massive number of new body types and fossil types that showed up at a certain period in time called the Cambrian era, and they couldn't have shown up via evolution because they all virtually appear overnight in the fossil record. They don't show any signs of having gradually been involved. And all of this culminated really as recently as 2016 at the Royal Society in London. This is the world's oldest scientific body. It was once chaired by Isaac Newton, and they held a conference in 2016 where the title was The Explanatory Deficits of Neo-Darwinism, or basically Darwinism cannot explain what it claims to explain, new forms of life having arisen. And, we just need a new theory, guys, because this one is not working, and we all know it. And one more. Come on, there we go. I'd like to end with this one, right? Something C.S. Lewis also said about science. Science works by experiments. It looks at how things behave. It says, here's what happened, but it can't tell you anything about the why behind it. That is something that's not a scientific question. The point being, you don't use science as a basis for your faith, but also don't use science as a reason for your lack of faith. It's a bad bet, worse than the Powerball lottery that we talked about last week. Let's continue.